Hello to all participants. I am Taha Altu from Kırıkkale University, Faculty of Veterinary Medicine, Physiology Department, and our study is about does different concentration of high fructose corn syrup impact some hematological parameters in broilers. And what is the high fructose corn syrup? Fructose is widely used as a sweetening substitute for glucose or sucrose in food processing and has become a major constituent of modern human diet. The excessive consumption in the world has resulted in the increased incidence of obesity, diabetes, and other cardiovascular diseases and metabolic syndromes. Health concerns regarding the consumption of HFCAs containing foods have developed in parallel with the increasing amount of people who become overweight as well. Corn is not only food for humans, but it is also feed for farm animals as diverse as cattle, pigs, and poultry that are the major source of meats. Corn is the primary source of high fructose corn syrup. Besides, byproducts derived from fruits have been used in poultry farms, and the acute effect of fructose on the chick's metabolism is still unclear. The aim of this study was to investigate the effect of high fructose corn syrup consumption on the hematological parameters in broiler. And our method, total 36 chicken broilers were separated into 3 groups with equal number of animals and fed ad libitum with isonitrogenic, isocaloric starter and finisher feeds and water for 42 days. Animals in one of these groups were given no other treatment as a control, while the diets of the broilers in the second and third groups contained 50 mg/kg or 100 mg/kg HFCAs. At the end of the experimental days, animals were killed by cervical dislocation and blood samples were collected in test tubes with anticoagulant for hematological analysis. There is no specifically a hemogram device for avian animals because of the avian bloods have a large nuclei. So, we have counted our parameters by manual methods. For the red blood cell, we mixed blood and a Hayem solution in Eppendorf tube. Then, fluid is allowed to slowly counting area, and we counted erythrocyte by using microscope. If we have counted N cells in one of the large squares, the concentration of our sample will be N multiply 10,000 cell divide milliliters. We used Drapkin's reagent to determination of blood hemoglobin levels. We mixed blood and reagent in the 96-well plate and displayed the results on a spectrophotometer at 540 nanometers. Packet cell volume. Taking blood in the microhematocrit tube, then one side of the hematocrit tube is sealed with paste, placed in a microhematocrit device to centrifuge 13,000 rpm for 5 minutes. Then the red lower half of the tube we have obtained packet cell volume. As you see, comparison with reference cover, it is about 42%. Those are also red blood cell index and how we calculate them, their formulas, mean corpuscular volume, mean corpuscular hemoglobin, and mean corpuscular hemoglobin concentration and their formula. Although it is very similar to red blood cell method, there are some differences. In red blood cell method, we have used Hayem solution and Thoma chamber. Also, dilution ratio was 1 to 200. In white blood cell method, we used Turk solution, Norbert chamber, and ratio is 1 to 10. Evaluating blood smear. We prepared a thin smear and air dry. and then we stained the smear in Megrunwald and Gimza. And we counted 100 leukocyte subtypes such as basophil, eosinophil, heterophil, lymphocyte and monocyte. Then we calculated the ratio of all of them. When we checked the results, hemoglobin levels of the broilers were not affected by fructose intake. And red blood cell was significantly higher in high dietary fructose group than low dietary fructose group. Fructose treatment significantly elevated the packet cell volume and mean corpuscular volume values of broilers in high dose groups rather than low dose group compared to control group. On the other hand, high fructose treatments remarkably reduced MCH and MCHC values compared to low fructose treatment and non-treated control groups in broilers respectively. And we checked the alterations in the leukocyte subtypes. Neither the lower nor higher fructose treatment were affected total white blood cell counts of broilers. However, percentage of basophil was significantly increased in animals treated low fructose, while it was similar to control in the animals treated high fructose. Effect of low or high fructose treatment on heterophil and lymphocytes were the opposite of each other. The lower fructose significantly increased heterophil and decreased lymphocyte, while the higher fructose decreased heterophil and increased lymphocyte values respectively. Monocyte didn't affect from the lower or higher fructose treatment. Heterophil lymphocyte ratio didn't change by high dietary fructose intake but significantly increased by low dietary fructose intake in broiler as seen table. In discussion, red blood cell and other parameters which are depend on the red blood cell count are observed significance. Packet cell volume and mean corpuscular volume showed difference related to red blood cell count as well. As a result, we thought HFCA's intake may trigger to bone marrow to produce erythrocyte. Also, the decreased MCH and MCHC may result from increased red blood cell production and volume without hemoglobin alteration. Although basically white blood cell count is not significant in no groups, some subtypes are showed significance, especially basophil and heterophil is increased in low fructose group. This situation is commonly result for an acute infection, allergic reaction, inflammatory disorders, stress, or chronic conditions. The heterophil lymphocyte ratio increases significantly with low dietary fructose intake in broilers. This result also confirms to us that the low fructose group may have some oxidative stress factors such as acute infection, allergic reaction, inflammatory disorders. In conclusion, this study revealed that dietary fructose intake may change some hematological parameters which can be first symptoms of further systemic or metabolic diseases. Also, future studies may focus on effect of HFCAs to blood cells in vitro and other species hemogram parameters. And those are some articles related to our study. Thank you for dear participants to listening.